Alright, now I'm going to come back to that protein a bit later on. What I'm going to do now is talk about a real protein. So this is ataxin-3, and this is something we've been working on for 8 years. Ataxin-3, we don't know what it looks like. We know what a bit of it looks like, but we don't know what the whole protein looks like yet. And what I just want you to remember about this protein is you can split it into two halves. Alright, there's the half that most people are interested in, which is the glutamines, and they're up here. And like Huntington's and like the ones I've told you about, if that polyglutamine tract is less than about 40, you don't see disease. Above 40 to 45, you get disease. The other half of the protein consists of something called Josephine, and that's a folded domain. Right? And that gives this protein its biological function. I'll come back to that in a moment. So what happens inside a cell if you have ataxin-3? Well, all of us have ataxin-3 present in all of our cells. It's present throughout the body. We don't yet know what the function of the protein is. Okay? We don't know what this protein does. All we know is it's present in almost every cell. And it sits there, here's a cell, and it sits there and is just spread throughout the cell. In this case, we've got ataxin-3 with 18 glutamine, so that's non-pathological, okay? And it's spread throughout the cell and it's doing whatever its biological function is. Over here, we've got cells which are expressing ataxin-3, but this time we've got 78 glutamines, we're in the pathological range, and what you can see, or I hope you can see, is within the cell, you can see these dark, dense spots, which they haven't come up too well. These are the aggregates that form, all right? This is this material, but within the cell. So all I want you to get from this is non-pathological, spread throughout the cell. Pathological, we form aggregates. All right? And it's believed that this is the stuff that's causing the damage. So I put this slide up, and I'm not really going to go through it, but I put it up every time because this is the slide that describes how we produce this protein in the laboratory. And the person who really did this is Michelle Chow, who's now moved on from my lab, but it took her probably 4 or 5 years just to be able to produce this protein reproducibly. It was a real feat, and without her being able to do that, the work I'm going to talk about just wouldn't have occurred. And she had a horror of a PhD in doing this. But what she was able to do is to establish a system where we can produce ataxin-3 with anything from no glutamines all the way up to 64 glutamines. All right? And what that's allowed us to do is to be able to study this. All right, so what I'm going to do now is tell you a story that probably took us about 4 years to work out. But we've now established how this protein aggregates. And the nice thing about this story is what we showed is no one believed us when we first showed it, but it's now been shown that it's the same for a couple of other scars and the same for Huntington's. So the mechanism I'm going to talk about is now looking like it's the standard mechanism for this family of protein. So what a student of mine did, Andrew Elston, was he took ataxin-3 with different glutamine lengths. So one with 2 glutamines, one with 15, one with 64. Remember, 64 pathological, 15 isn't. And he did the same experiment Amy did. He just put it in a test tube and watched it. And he waited for that protein to aggregate. And when it aggregates, we're able to see that. All right, here you've got a blot. And all you need to know really is where you see these bands forming, that's where we're finding aggregates. So he left the protein in the test tube for a while, and at various time points he took the protein out and put them on this paper. All right? And then we're able to detect aggregates. And what you can see is that the ataxin with 64 repeats begins to form these aggregates quite quickly within 8 hours, 23 hours. We're working at very high concentrations, far higher than you'll ever find in the cell. So this process happens quite quickly. And you can see by 32 hours, all of the protein has been converted to this form. Whereas you can see that if you've got 2 glutamines or 15 glutamines, nothing happens. The protein just sits there in the test tube, doesn't change shape, doesn't do anything. All right? And that's why we don't see any bands. So what this tells us is, at the molecular level, we know that once you've got more than 40 or so glutamines in a row, ataxin-3 will aggregate. And we can follow this reaction in many different ways. What do these aggregates look like as this reaction's occurring? What you can see here are 3 electron micrographs of ataxin-3 with 64 repeats at various time points. So again, same experiment. They were just put in a test tube, and he took them out and then looked at them under the electron microscope. What you can see, after 10 hours, we begin to form these small worm-like species. Within the field, they're called protofibrils. And these things get bigger, and they form longer worm-like species. And then over time, we form these species, which are fibrils, which actually then literally wrap around each other like rope. So you go from forming these small sort of tangles to forming these big ribbon-like, big rope-like structures. What's important in this reaction is we're able to take samples out and we're able to look and see if things that are being formed are actually toxic. So a student of mine, Bronwyn Thomas, was able to reproduce this sort of experiment, and she took aggregates at different time points. And what she then asked the question was, do these aggregates, these fibrils, do any damage to cells? So in the experiment she just had cells, she added the aggregates to them, and she asked, did the cells survive? And what she found is that the aggregates that form at around 60 hours in this reaction, so that's these species here, are actually highly toxic to cells. Whereas aggregates that are formed very early or very late actually don't do anything. So this gives us clues as to how this protein, when it aggregates, begins to cause disease. We now know which type of molecule or which molecular species is presumably causing damage to the cells. And in knowing this, we can begin to hopefully try and stop it. So we did a huge amount of work. Well, I didn't. The people at the back did the work. In trying to understand how this protein, which is normally soluble, all right, just sits there, doesn't do anything, undergoes this change to form this mess that we see over here. And through that, we discovered a few interesting things. And I'm just going to show you in diagrammatic form what happens. So this is our protein. This is ataxin 3. If you remember, I told you it's a protein of two halves. All right. In red, you've got the polyglutamine tract. In green, we've got that other thing, Josephine. All right. What Andy and a couple of others in the lab showed is that when this protein changes shape, now we don't know precisely what that change in shape is. It forms those worm-like species. All right, and that I've represented here. What we know is that that initial change in shape is actually not because of the glutamines. It's actually because of the Josephine. All right, so this is where the story gets a little bit confusing. All right, the Josephine itself changes shape, which is why I've now got this thing in blue. The glutamines are now hanging there. What happens, though, is because the Josephine changes shape, it comes together. It actually brings the glutamines, polyQs here, close enough together that they can then interact. And when they interact, that's when we form these things. All right, so this is what we call a multi-domain mechanism. When we presented this, well, actually, when Andy came and presented it to me, I refused to believe him, because it goes against all the dogma in the field. Up until this experiment, everyone thought it was only the glutamines that were really important. And they are incredibly important, but they're not the whole story. The work that Andy did and others in the lab showed that it's actually this, the Josephine domain which starts this off, and then the glutamines kick in. Something similar has been shown for SCAR1, something similar has now been shown for Huntington's. So it looks like we were the first in getting to the molecular mechanism by which this actually occurs. Alright, so when we presented this, we just got laughed at. No one believed it. We had to go away and test it and prove it, which we did. And there's one experiment I'm going to show you because it's actually important for the last sort of part of my talk. What that slide in very big writing says is if it's Josephine, this thing here that has to change shape for this protein to aggregate, if we change how that protein changes shape, if we can prevent it, then presumably we can stop this protein from aggregating. All right? If we can stop this, then we should be able to stop it. So in the laboratory, that's fairly easy to do. What we do is we change the amino acid sequence. All right? And what we did is we put mutations into the sequence which make Josephine really stable. So it can't change. All right? It forms its structure and it's stuck there like a rock. So we did that and we asked the question, if we put these mutations in which make the protein stable, can we prevent this reaction? And the simple answer to this is yes, we can. All right, so over here is the structure of Josephine. A group in London actually beat us to the structure. They solved it, so we know what it looks like. And what we did is we introduced a mutation over here which stabilised the protein. All right, so all that means is if we heat this protein up, all proteins begin to change shape. By stabilising it, that occurs at a much higher temperature. The protein's like a rock. All right? And what you can see down here, or I hope you can see,